Hi, everybody. I'm Ethan Perlstein, the founder and CEO of Perlara and the co-founder and CEO of Maggie's Pearl. I'd like to share with you a story that I recently told to folks live in San Diego recently at the CDG Care Conference, and I'm recording this video here as a redo of that talk. So I'm going to share my screen and also start the slide share. And the title of my talk is Epalrestat, the first drug that normalizes PMM2 CDG disease measures in several pioneer studies. Basically, I'll tell you a story of how we did drug repurposing using simple animal models like yeast and worms, and went from fibros to kiddos in a series of steps that I'll describe here. And then I'll end with a preview of what's to come next. So this journey began courtesy of a pioneer, a PMM2 CDG pioneer named Maggie from Michigan. And she has the most common congenital disorder of glycosylation, or CDG, called PMM2 CDG, or CDG1A once upon a time. PMM2 CDG affects over 1,000 kids globally, 1,000 patients globally, most of them kids like Maggie. And at the start of 2020, before the pandemic, Maggie began taking a repurposed drug called Epalrestat, and we discovered this drug together, and that's what I'll describe. And we use these simple patient avatars to do so. And what I mean by it's working is a combination of real-world evidence and some measures that we've published based on Maggie's N of 1 study. So this is a piece of real-world evidence. This is a walking compilation. So this is Maggie from May of 2019. This is, let me turn off the sound here. This is from May of 2019. And you can see how she got around using this assisted walker setup. Here's August of 2019. So this is about 5 months before the beginning on Epalrestat. This is after Maggie, right before I think Maggie started taking acetazolamide. And you can see still here that she's having difficulty walking in a straight line and maintaining a straight line. I'll advance a little bit to, let me go back a little bit here to the transition. So this is a couple of months on Epalrestat, in the same walker, and I think you can already start to notice a difference in her gait, more ability to hold a line and to be able to make this turn, relatively sharp turn into the elevator and then turn around. And then here's 6 months on Epalrestat. I think you can see more confidence in each of the strides there, navigating over this little obstacle there, and then getting back onto the flat surface here and kind of picking up some steam. And there's a couple of segments like this. This is October of 2020, also navigating here a bit of an obstacle course through Mayo waiting room area, paying attention, I think, to her footsteps, and maybe noticing again sort of more stability and more precision with each step. And then here's a sequence from the last summer, outdoor sequence here on kind of this rubbery surface here on this outdoor court, outdoor play area. And then we'll kind of wrap up here with a sequence with some assisted walking, actually, walking back and forth across sort of a room in Maggie's home. So kind of let the real-world evidence do the talking here, and then Maggie will do a turn and we'll walk back. And we've accumulated obviously more videos of this sort since, but this kind of constitutes how far she's come on her N of 1 journey. And we'll get to some of the other PMM2 pioneers and introduce some of the real evidence from their N of 1 studies. But I'll now advance to, here's just sort of a sequence I can go a little bit faster through. This is to illustrate changes in her core stability So this is not involving locomotion, but now seated. But you can see here, this is now November 18. And this will be a sequence will show kind of the same couch. It'll be a different angle, a camera angle, but you can notice here that she's having a hard time kind of sitting still and jerkiness to the movement. This is sort of classic presentation here. And then we'll skip ahead to May 2020. So this is a period about 5 months or so on Epalrestat treatment. So coinciding with some of the sequence from the walking video. And you can see here, I think, the improvement in stability. And then this will kind of cycle through a number of segments. And here, sitting on this ottoman without any sort of support behind her. And then being able to extend her arms out and then touch her nose. And then here's sort of a sequence of kind of doing, getting up on her knees there and maintaining kind of this core stability. And there will be other videos that have been made since, but this kind of again documents the steps along her or Maggie's N-of-1 journey so far. And then here's some evidence, real evidence in the form of fine motor improvements pre and post Epalrestat with coloring, with tracing or drawing her name. So how do we get to a point where we could identify a compound, a repurposed drug that had been out there in the world, specifically over in Asia, but this molecule existed out in the world, but we didn't know that it had potentially beneficial effects in this rare metabolic disorder. The connection was made courtesy of these so-called patient avatars, these yeast and worm and fibroblasts, patient-derived fibroblast models that we could use to perform a set of drug repurposing screens that would give us recommendations about what strategies to use, what therapeutic strategies, what mechanisms, and what potential even specific molecules could be tested. And then, of course, the sequence goes from fibros to kiddos. And that's kind of I give you a little bit of a teaser of where we're going here. But now I want to retrace our steps back to how this was made possible. And so with yeast models, and I'll come back to them at the end, we were able to exploit different kinds of variants or mutations that cause PMM2. And they break the PMM2 enzyme in different ways. And we could basically show that in a simplified yeast system, where we're just measuring the growth of a cell, we could show that these different variants caused a more or less severe growth defect. And then, of course, we could try to find drugs that rescued that growth defect. We went through that exercise and we found a number of molecules that kind of didn't really have any clinical promise per se, but they showed us and they taught us that we could identify mechanisms that worked in one mutation, one variant, one flavor, red, but work more preferably there over the purple mutation or the purple variant. And we could find a situation that was flipped. And then we could find an example of a molecule that would rescue the growth of these two different PMM2 variants equally. All that to say that this yeast model really generated a few leads, and those leads then we built upon using a worm model. And this just shows you that if you measure the enzyme activity of PMM2 in this F119L homozygote model, we could show a decrease in activity relative to a control. And that showed us that we had a bona fide model. Then we can visually see how PMM2 expresses itself in worms by the fact that they don't really grow well. And on the left is a picture of a well or a field of worms that have PMM2, this mutation installed in it. We've stressed them a little bit more with another molecule called bortezomib, but that detail is not really important for the purposes of this discussion. And we could look for drugs, just like we did in the case of yeast. We could look for drugs, repurposable drugs, that could make the PMM2 worms healthy. And an example of what that would look like is the thriving population of worms on the right. And we found a list of molecules summarized here, all published work. And what I'll just sort of skip to the chase here is that there was a yeast molecule, a yeast hit, And it looked a lot sort of like a worm hit. And then that led us to a known molecule, a known actual manufactured drug called Epalrestat, or Kinadac in Japan. And we were able to make this series of connections courtesy of the patient avatars and putting on our thinking caps and looking at structures and making these organic intelligence connections as well. And this just summarizes that the cinnamon compound on the left, which we first identified using the yeast PM2 models, it ended up checking out and working in the worm as well, and working in fibros. And then we found that Epalrestat, which is a chemical version of this compound that's synthetically derived, so not from nature, but by chemists from Ono, had very similar properties. And this kind of summarizes the boost in PM2 enzyme activity that we observed in the Epalrestat condition and in the CHCA, which is the cinnamon compound treatment, in both worms on the left and in human fibros on the right. And here's a series of experiments that just went to show that we could see this boost of PM2 enzyme activity across a number of different patient samples. We could show that the actual proteins inside the fibroblasts of PM2 patient cells, we could take their glycosylation defect, the fact that they have less glycosylation, less sugar attached to them, we could fix that. And this series of volcano plots shows that, basically all the points going from the upper left to the upper right. And then we also could show that based on what we knew of Epalrestat's effects in adults who take it for the treatment of peripheral neuropathy of their diabetes as a complication of their diabetes, we knew kind of how quickly or what the profile of Epalrestat looked like in adult blood. And we could show that in Maggie's case that the so-called PK or pharmacokinetic profile of Epalrestat is basically the same, whether this is a pediatric or in the previous case, the adult setting. And then we also were able to take advantage of the history of the Epalrestat and its use in diabetic neuropathy and to kind of spark an insight, which is that there was an increase in sorbitol, in urine sorbitol specifically, noted in diabetic patients versus non-diabetic patients, and Epalrestat could fix that. And so we wondered, could that also be the case in this glycosylation disorder, where there's an excess in buildup of glucose in cells, not because of excess in diet, but because of this inability to glycosylate proteins. And indeed, there was a buildup of both sorbitol and mannitol in Maggie's urine, and Epalrestat treatment could normalize that. And this is part of the published study from last fall with the FCDGC, showing that that polyol levels, sorbitol and mannitol levels, are increased in a number of patients and in PM2 patients, and it's correlated with neuropathy and liver pathology scores. This is just summarizing that Maggie's ICARS, which is a functional test of ataxia, she showed improvement. Lower numbers are better in this score. And she had started improving on acetazolamide, washed out of acetazolamide, and then went on Epalrestat, continued her trajectory of improvement, which would not be expected if she had been withdrawn if she had been cleared of acetazolamide. Normally she would have reverted back to her baseline, but instead she actually continued to improve, and all the while her liver enzymes were stable. And I'll just sort of wrap up here with a video of the second PMM2 child who started on Epalrestat. These were his first unassisted steps. 3 months of treatment, 4 months of treatment. You can see here again, these are his first unassisted steps. And his distance traveled gets a little bit longer, the longer he's on Epalrestat. And here's the last in this segment. Able to take 5 steps there. And what I want to kind of conclude on here is that we were able to take advantage of these yeast patient avatars and look for known drugs that could rescue their growth defect. And then we wondered, could we do another type of experiment, which is take those same yeast avatars and let evolution or let nature tell us what changes or what mutations need to happen to make the cells healthy again. And so we did this so-called therapeutic evolution experiment, which is now available on bioRxiv as a preprint and has been submitted for peer review at an open access journal. And so stay tuned for that, but you can read all of this as well just as well in the preprint on bioRxiv. But we were able to evolve 2 different flavors of PMM2 mutant yeast over time. And we're able to show here that we could actually, these little traces show little independent populations of these sick PMM2 yeast evolving and gaining growth improvement over time. And that's what that means when they're sort of go up and to the right in these graphs. And this just sort of summarizes that we were very shocked to find that the mutations that accumulate in these PMM2 yeast that evolve to get healthier, the mutations that accumulate are in actually other CDG genes, other genes that can cause congenital disorder of glycosylation, including one gene in particular, PGM1, or phosphoglucomutase. Remember that PMM2 is phosphonanomutase deficiency. There's another CDG called phosphoglucomutase deficiency. And it turned out mutations in PGM1 seem to make PMM2 yeast better. And that was really a startling finding. And we were able to collaborate with a biochemistry lab, Professor Andreotti's lab in Italy, that was able to look at the effects of the different mutations that arose by therapeutic evolution and assess what did the mutations do to PGM1. And they turned down the activity of the PGM1 enzyme. And we can kind of show here this really interesting finding where if you compare the amount of enzyme activity of PGM1 enzyme activity that you dial down, and how does that compare to how much increase you get in your PMM2 yeast mutant, there seems to be this sweet spot of reduction of PGM1 enzyme to about 25% of normal seemed to cause the greatest increase in our PMM2 yeast. So it suggested that that one CDG gene could be modifying another, and that this modifier relationship is maybe very sensitive to exactly how much you're tuning one gene's activity relative to the other. So this is all work that's being now submitted for publication, but is available by bioRxiv. And the original work and all of the N of 1 Epalrestat study with Maggie has been done with Dr. Eva Morava-Kozic. So thank you all for your time here, and hope to see you next time at the conference in San Diego for the next CDG Care meeting. Thanks, everybody.